Hi everybody. Today we shall discuss some of the questions from molecular basis of inheritance up to packing of genetic material. Let us see some of the questions from this particular chapter. Molecular basis of inheritance, the first part. Let us see the first question. Choose the correct statement. DNA is the genetic material in all cells that follow cell theory. Yes, absolutely true. It is genetic material in all the cells that follow cell theory. They are both prokaryotes and eukaryotes. It is only the DNA that acts as genetic material. In all viruses, DNA is the genetic material. It is wrong. Only in some viruses, DNA is genetic material. In some other viruses, RNA is the genetic material. DNA is the genetic material in viroids. No, RNA is the genetic material in viroids. RNA is the genetic material in prions. No, prions do not have DNA and RNA. They have only protein, that is misfolded protein. So first option must be the correct one. Let us see the answer. First one is given. In all organisms that follow cell theory, both prokaryotes and eukaryotes, DNA is the genetic material. Viruses have either DNA or RNA as genetic material. Viroids have RNA as genetic material. Prions have neither RNA nor DNA as genetic material, and they are made of misfolded proteins. Coming to the next question. The matching type. So this is given from the introductory part of this chapter. The acts as messenger. So for carrying the genetic information, so one of the nucleic acids acts as messenger, that is messenger RNA, mRNA, A4, acts as adapter. Transfer RNA acts as adapter between the messenger RNA as well as the amino acids involved in protein synthesis. Transfer RNA carries the amino acids onto the site of ribosomes. Right? It has a structural role. Only it has got structural role, that is 16S ribosomal RNA has got only structural role. It is present in 30S subunit of 70S type of ribosomes. Ribosomal RNA has a structural and catalytic role. 23S rRNA, that means ribosomal RNA, it has structural role being a part of larger subunit of 70S type of ribosomes, that is 50S subunit, and also it acts as an enzyme peptidyl transferase which is involved in peptide bond formation. So B1, so A 4, B 3, C 2, D 1. So second option must be the correct one. Let us see the answer. Second one. Solution. 23S ribosomal RNA or 70S ribosomes has structural role and catalytic role as peptidyl transferase. 16S ribosomal RNA has a structural role as part of 30S subunit of 70S ribosomes. Messenger RNA acts as messenger and carries protein synthesis information from DNA to cytoplasm. Transfer RNA acts as adapter between amino acids and messenger RNA bound to ribosome during protein synthesis. Coming to the next question. Which is the main difference between DNA and RNA? So there are several differences between DNA and RNA, but the main difference by which it is described as a DNA or RNA is a type of the sugar. That is, DNA has got deoxyribose sugar, RNA has got ribose sugar. The other features, whatever may be the other features, the sugar is deoxyribose, it is DNA, and the sugar is ribose sugar in the nucleic acid, it is RNA. Let us see the options. RNA is a single stranded and DNA is double stranded. No, RNA is usually single stranded and DNA is usually double stranded. Sometimes DNA is a single stranded and RNA may be double stranded as in certain viruses. Purines and pyrimidines do not exist in 1:1 ratio in RNA, whereas it does exist in DNA. Only in double-stranded RNA and DNA, purines and pyrimidines exist in 1:1 ratio. The DNA is single-stranded, purines and pyrimidines do not exist in 1:1 ratio. So, second option must be the wrong one. RNA has uracil and DNA cytosine. Cytosine is present in both DNA and RNA, but RNA usually has got uracil and DNA has got thymine. But in some cases, certain viruses, so certain viruses have got even thymine in its RNA, uracil in its DNA, right? So that is also not the main one. RNA has ribose sugar and DNA deoxyribose. This is the main one. So this is the main one by which the nucleic acid is described as either DNA or RNA. Fourth option must be the correct one. Let us see the answer. Fourth one. The main difference between RNA and DNA is the type of sugar. RNA has ribose sugar and DNA has deoxyribose sugar. RNA has uracil and DNA instead of uracil usually has thymine. But there are certain viruses, so DNA viruses, uracil may be present, such as PBS1 and PBS2 viruses. RNA is usually single-stranded, yes, and DNA double-stranded. But there are cases where the RNA is double-stranded in some viruses and DNA is single-stranded in some viruses. If single-stranded, purines and pyrimidines do not exist in 1:1 ratio in both DNA and RNA. If double-stranded, then only in RNA and DNA, purines and pyrimidines exist in 1:1 ratio. Coming to the next question. Statement 1, Statement 2. In a nucleotide, the phosphate is esterified to sugar by 5' phosphoester linkage. Yes, absolutely true. The phosphate is attached to the fifth carbon of the ribose or deoxyribose sugar of a nucleotide. Absolutely true. N-glycosidic bond is formed between sugar and nitrogen base of nucleotide of nucleic acid. Yes. So, the N-glycosidic bond is formed between the first carbon of the ribose or deoxyribose sugar and to the nitrogen present at the first position or the ninth position. First position of the pyrimidine and the ninth position of purine in case of nucleotide as well as nucleoside. The N-glycosidic bond is formed between sugar and nitrogen base in case of both nucleotide as well as nucleoside. Whereas phosphoester bond is present only in nucleotide but not in nucleoside. So statement 1 and statement 2 both are correct. Both statement 1 and statement 2 are correct. Fourth option must be the correct one. Let us see the answer. Fourth one. In a nucleotide, the phosphate is attached to the fifth carbon of the sugar. 5 fifth carbon of the sugar by phosphoester linkage, and the nitrogen base to the first carbon of sugar by N-glycosidic bond. Between adjacent nucleotides of the same strand, there is establishment of 3' 5' phosphodiester linkage. Hydrogen bonds are formed between complementary nitrogen bases of opposing or same strand. When the same strand is folded, when complementary bases come opposite to each other same strand, then also there is a formation of hydrogen bonds. They may be found between AT or AU in case of RNA and JC in case of both RNA and DNA. Coming to the next question. Frederick Michel. Frederick Michel discovered DNA as acidic substance in the nucleus under the name nucleins. The acidic nature of nucleic acids, particularly DNA, was discovered by Frederick Michel. So, in the nuclei of the cells, Rosalind Franklin and Morris Wilkins both gave X-ray diffraction data on DNA. So, A, 2, B, 3. Erwin Chargaff described that the ratio between adenine and thymine of double-stranded DNA is equal. Amount of adenine is equal to the amount of thymine in DNA. Amount of guanine is equivalent to amount of cytosine. So we also concluded that amount of purines is equivalent to the amount of pyrimidines. So C4. Watson and Crick gave the double helical model for DNA. So A should be the 2, A2, B 3, C 4, D 1. So A2, B3, C4, D1. Third option must be the correct one. Let us see the answer. Third one is given. Frederick Michel. DNA as acidic substance of nucleus. Rosalind Franklin X-ray diffraction data on DNA along with Morris Wilkins. Erwin Chergaff, existence of adenine and thymine and guanine and cytosine equal proportions in double-stranded DNA. Watson and Crick gave double helical model for structure of DNA. Coming to the next question. Choose the incorrect statement with respect to DNA structure as given by Watson and Crick. Incorrect one here to choose. The sequence of nucleotides of one strand of DNA is known. The sequence in another strand can be predicted. Absolutely true, because the sequence of the nitrogen bases or nucleotides of one strand is complementary to another one. So naturally it can be predicted. Both strands of DNA show parallel polarity, no, anti-parallel polarity. One strand is directed toward 5 prime, 3 prime, and other strand 3 prime, 5 prime polarity. So it is anti-parallel, not parallel, anti-parallel. Second option must be the correct one. That is the incorrect statement. The bases of opposing strands are paired through hydrogen bonds. Absolutely true. Adenine pairs with thymine with 2 hydrogen bonds between them. Guanine pairs with cytosine with the 3 hydrogen bonds between them. The two strands are coiled in right-handed direction, absolutely true, clockwise direction. And the pitch of helix is 3.4 nanometers or 34 angstroms. So the second option must be the incorrect statement. So let us see the answer. Second one. The opposing strands of DNA show anti-parallel polarity. One strand has 5 prime 3 prime polarity, and another one has 3 prime 5 prime polarity. Coming to the next question. So assertion and reason type question this is. The daughter DNA molecule is formed from parental DNA molecule not only identical to each other but also identical to the parental DNA in their nucleotide nitrogen base sequence. Absolutely true. So, because during DNA formation, both the strands of DNA act as template, and in both the template DNA strands, a new strand is formed, right, whose sequence is complementary to the sequence of template strand. Since both the template strands are complementary, even both the strands of both the DNA molecules are also complementary in terms of nitrogen base sequence. So, both the two daughter DNA molecules exactly resemble the parental DNA molecule. The sequence of bases of one strand of DNA is complementary to the sequence in opposing strand. So it is also true. Reason is also correct. Assertion is also correct. Reason explaining the assertion. So A and R are correct, and R is the correct explanation of A. Second option must be the correct one. Let us see the answer. Second one. The sequence of bases of opposing strands of double helical DNA is complementary, and both the strands of DNA act as templates. The two daughter DNA molecules formed are exact copies of the parental DNA molecule with the same sequence of nitrogen bases. Coming to the next question. Central dogma of molecular biology indicates the flow of genetic information as, according to central dogma of molecular biology given by Crick, the flow of genetic information is from DNA to RNA, that is messenger RNA, and from RNA to protein, DNA to RNA, RNA to protein. He also shown that from DNA there is also formation of DNA. Formation of DNA from the existing DNA or parental DNA is DNA replication. Formation of RNA from DNA is the transcription, and formation of protein on the surface of ribosome as per the information present on messenger RNA is described as the translation. So first option must be the correct one. Second option is not correct. RNA to DNA wrong. It is DNA to RNA. RNA to RNA is wrong. It is DNA to RNA and protein. Protein to RNA it is wrong. So first option must be the correct one. First one. Here is the answer. The Crick represented the central dogma of molecular biology to show the flow of genetic information as this one. Very clear. From DNA to DNA replication process, DNA to RNA transcription process, and RNA to protein, that is a translation process. Coming to the next question. Identify the correct statements. The DNA of diploid cell of man has a length of approximately 2.2 meters. Absolutely true. It is given in the book. The first year book it is given as 2 meters. In the chromosomes, in the nucleus, but here 2.2, 2 point exactly it is correct. 2.2 meters. So each haploid cell DNA has got 1.1 meters, diploid cell 2.2 meters. Absolutely true. The length of equinoid DNA 1.36 millimeters. The number of base pairs in it is absolutely true. 1.36 into 10 to the power of 6 by 0.34. 0.34 because it is the length of each pair of nucleotide is 0.34 nanometers. So 1.3 millimeters converted to nanometers by multiplying with 1 million, that 10 to the power of 6. That is 4 into 10 to the power of 6, that is 4 million. Right? So 4 million bases, 4 into 10 to the power of 6, that is also correct. In prokaryote, DNA is held by some positively charged proteins in the region of nucleus. So in the prokaryotic cell, DNA is not present throughout the cytoplasm. It is limited to a certain region of the cytoplasm because the DNA is held by some positively charged proteins. That particular region where the DNA is held by positively charged proteins is known as nucleoid. Histones are basic proteins held to DNA of eukaryote. Absolutely true. Histones are basic proteins and they are held to the acidic DNA or negatively charged DNA. This is also correct. Nucleosome of eukaryotic chromatin is formed due to wrapping of DNA around histone octamer. Yes. Each nucleosome has got a core of histones, eight histone molecules belonging to four types: H2A, H2B, H3, and H4. Each one two copies. And those core histones are wrapped by DNA almost twice to form a nucleosome. So E is also correct. A is correct, B is correct, C is correct, D is correct, E is also correct. A, B, C, D, E are correct. First option is the correct one. Let us see the answer. First one. Length of DNA of human diploid cell is 2.2 meters. Number of base pairs in equal DNA is 4 millions. In prokaryotes, the DNA is held by positively charged proteins in the nucleoid region. In eukaryotes, the basic proteins organized as octamer of histones is wrapped by DNA to form nucleosome organization of chromatin. Coming to the next question. Highest level of DNA packing in eukaryotes is seen at. So, greatest degree of packing is seen where the chromatin is the maximum condensed state. In eukaryotes, chromosomes are in maximum condensed state in metaphase. Metaphase must be the stage where highest level of DNA packing is seen. Not telophase, telophase relaxed. Prophase, the condensing stage, that is. So condensation is in process in prophase. By metaphase, it is complete. Interphase, highly dispersed, the chromatin is. So metaphase is the most correct one. So first option must be the correct one. Let us see the answer. Fourth one. Less packing of DNA of eukaryotes is seen in interphase and highest level is seen in metaphase, where the chromosomes are in maximum condensed stage. Both histones and non-histone chromosome proteins play a role in packing of DNA of eukaryotes. So, all about, please like, comment, subscribe. Thank you.